Hi everyone, and welcome to the last seminar before the holidays. And we take great pleasure in having Dr. Lauren O'Donnell as our speaker today. Lauren is an associate professor in radiology at Harvard Medical School and Brigham and Women's Hospital, and her research focuses on developing novel computational tools for the analysis of diffusion MRI. She received her BS from Case Western, an MS in CS from MIT, and then went on to get a PhD from this joint Harvard-MIT Health Sciences and Technology program. I think it's a joint between the Medical Vision Group and CSAIL, Computer Science and Artificial Intelligence Laboratory at MIT, and from Eric Grimson's group, who is very, very famous in the field. Lauren closely collaborates with the folks from Golby Lab, who do image-guided neurosurgery, the Laboratory of Mathematics and Imaging at Harvard, and the Surgical Planning Lab. And she's made key both methodological and software developments of several open source packages, one of them being Slicer, which is widely used by the surgical planning community. And she's very much also actively involved with the MICI community and the MICI Computational Diffusion MRI group, which hosts workshops every year. And she delivered a keynote lecture for 2021 this year. So today she's going to talk about two things. One, mapping these deep white matter fiber tracts in very large harmonized diffusion MRI datasets. A topic which is very relevant now, especially with the public release of large brain databases. And two, mapping the superficial white matter, these small fiber pathways near the cortex, which are now beginning to be studied as more high resolution data sets become available. So let's welcome Lauren.
Thank you very much. And
I also see Carl Frederick Westing here, so welcome, Carl.
Oh, okay, my CF, yeah. Hi, everyone. Thank you for joining. Thanks for coming to see my talk. So I'll be speaking today about mapping the human brain's deep and superficial white matter. And I'm a little bit jokingly referring to that as big data and small data. So what we're interested in, at least what I'm interested in here, is studying the white matter fiber tracts, or the connections in the brain, especially the human brain. And you can see here in two colors, this is an elaborate anatomical dissection of a brain. And you can see in two colors, the yellow color represents the arcuate fasciculus, and that's one of the deep white matter fiber tracts. And then you can see a magenta color that's highlighting a superficial white matter fiber connection. And so overall, all of these deep and superficial connections combine to form the brain's wiring diagram. And diffusion MRI is the only method that we have that can measure these brain connections in vivo. So to understand what they look like in living humans, and of course, we have no interest or ability to do brain dissections, which you see on the left. This is tracts that we are interested in, but we want to study these in living humans to see how they are in health and disease, what's their variability in the population. So then we have, as you see in the middle, diffusion MRI. And that works on a basic principle of diffusion anisotropy. There's a citation on the very bottom of the screen. That's a really nice citation that explains the basis of the diffusion anisotropy in the nervous system. But fundamentally, we're able to measure slower water diffusion across a fiber and faster diffusion along a fiber, where a fiber is a bunch of axons running parallel to each other closely together. Now, this is imperfect, but we can do mathematical modeling and computational processing, and we can estimate the true underlying fiber tracts. So we're interested in the fiber tracts you see on the left, and what we have is what you see on the right, a process called tractography that enables us to estimate their locations and trajectories in the brain. And so tractography plays a unique role. It's the only method that allows us to trace the connections in the brain. So we can trace and identify white matter connections or fiber tracts. This is used for visualization. It's used clinically for surgical planning. We can also try to quantify and measure information from these connections to study the brain. We can study their tissue microstructure from diffusion MRI information. We could study their volume. A lot of people study their structural connectivity, quote, strength, which is the strength of the connection between two cortical regions, and that can be defined in many ways. But overall, this allows us to study white matter fiber tract anatomy and/or the brain as a network in health and disease. So I'll be talking here about some unique opportunities that we can study now using tractography. The left illustration is intended to represent an example deep white matter tract, and we're able to study these on a large scale in new open large data sets that are coming out. And on the right, this is intended to represent an example superficial white matter tract. And we can study those to some extent in these large data sets. And then I'll talk later about some new data sets that are higher resolution that will allow us to see superficial white matter connections that we couldn't previously see, and how we plan to study those. So overall in my talk, I'll first talk about deep versus superficial white matter, an introduction, and then I'll talk about our two R1 projects right now in my laboratory. The first one is about studying especially the deep white matter using big data sets, large open data sets. And I'll talk about data harmonization of approximately 10,000 scans and our current results there. And then I'll talk about our superficial white matter project that just started this year. And I'll explain a bit about what is the current state of the art with superficial white matter tractography and what progress are we trying to make. So these are two images on the left and the right of the deep white matter anatomy using tractography. So the deep white matter anatomy, we really know a lot about it. Tractography is imperfect. There are many citations, many papers that can explain that there are certain biases and there can be false positives, et cetera. But at a fundamental level, we're really able to trace and identify these deep white matter tracts in a really robust way in large populations of subjects. These images are from two different computational methods that can automatically extract especially deep white matter fiber tracts. And ours is on the right. It's based on an atlas using machine learning fiber clustering. And I'll be describing that a little bit more in future slides about the deep white matter and also the sort of long range superficial white matter connections in our current atlas. So why do we care about these automatically identifying these fiber tracts or parcellating the deep white matter? We care about it in large part because it enables research to understand the brain. So this is a paper from my PhD student. You see her picture on the right, Leo Zeckelman. And one of her recent findings that's about to be published in NeuroImage, it was just accepted, is that the number of streamlines, which is an example piece of data that you can measure from a fiber tract, that it influences performance on a semantic memory task. So this is really interesting because you can look where in the brain does the microstructure or other properties of the fiber tracts, where in the brain does that basically relate to task performance. And that starts to give you a sense of what are the underlying structural networks that are related to that task. In this case, it's a language related task. And so it's interesting, as you would expect, you find more tracts in the left hemisphere that are related to the semantic memory performance than you find in the right hemisphere, because we know that in the majority of people, language is lateralized to the left hemisphere. So there's a lot of research that the identification of fiber tracts can enable. Now, what do we know in contrast about superficial white matter? This is the second part of the introduction, the part about superficial white matter. We know that the total volume of the U fibers, which are the superficial white matter connections that connect neighboring gyri and nearby gyri, these U fibers, their total volume is estimated at 240 centimeters cubed. And that is actually twice the volume of the deep white matter. So it's kind of amazing because the cortex is so highly folded. The layer of superficial white matter immediately under the cortex is actually very large, has a very high number of connections. And this is thought to enable, the superficial white matter is thought to enable cortical communication, which makes perfect sense because it connects neighboring and nearby cortical regions. And on this image on the left, you can see some of the U fibers in blue. You can see many U shapes connecting neighboring gyri and nearby gyri. So what do we know now about superficial white matter anatomy using diffusion MRI and tractography? Well, there was a really nice review paper that you can see in the lower left. Hopefully it shows on your screen in NeuroImage in 2020. called Superficial White Matter, a review on the diffusion MRI analysis methods and applications. This is a really nice paper that describes what is the current state of the art today regarding superficial white matter mapping, especially with tractography. And one of the things that this paper talks about is existing superficial white matter atlases. Now, this picture I'm showing here is part of a figure from that paper. And the figure from the paper aims to show similarities and differences between existing superficial white matter atlases. So B is an example of some basically connections in one particular superficial white matter atlas. And then below it, the bottom row, those are actually connections from our atlas, the superficial white matter connections that are in our atlas that weren't present in any of the atlases in previous rows. So you can see right now, there's a lot of differences across these different atlases. And however, you can also see that there's a lot that we can already describe basically and identify in the superficial white matter, even with current diffusion MRI. So far, the deep white matter has been the main focus of diffusion MRI research. So from the very beginning, Carlo Pierpaoli's paper, Diffusion Tensor MR Imaging of the Human Brain, which is a very famous radiology paper from 1996. So at the very beginning of diffusion MRI, we looked at white matter and gray matter and CSF, and they looked at the trace, which now is proportional to the mean diffusivity, and they also looked at an anisotropy measure that you see on the y-axis, and you see the trace on the x-axis. And there's this division, this really nice classic division of anisotropy greater than 0.2 is the white matter. But this is mostly focused on the deep white matter that you could really actually see with an anisotropy above 0.2. And neuroanatomical research has also primarily focused on the deep white matter. And this is a picture over here on the right from the Fiber Pathways of the Brain book. And this is a tracer injection into the motor cortex of a rhesus monkey. And you can see the injection site because it's very bright. And they call that in the book the injection halo. What you can really immediately notice about the injection halo is that it obscures the nearby local superficial white matter connections. So those are already harder to see. And then what they write in the book about these local connections, they write that the local association fibers provide diffuse terminations. So even in our source of ground truth about connectivity data, which is existing monkey tracer studies, we don't know that much about the superficial white matter. The studies people really need to go back and to look at these studies, which is something we're doing with a collaboration with neuroanatomists as one of our projects, to basically take a look at these studies, and they're trying to better understand what they tell us about the superficial white matter, because that was not the focus of these existing studies. And then in the lower left, you can see a quote from neuroanatomist and NeuroImage: white matter has been structurally defined in the past only at its core regions or its deep white matter. So all of these superficial connections that we can see with tractography, we don't really have names for them, like the arcuate fasciculus or the corticospinal tract, like we do with the deep white matter and the deep white matter tracts. So what about tractography research? We talked about general diffusion MRI research, and then we talked a bit about neuroanatomy research, but does tractography research really focus on the deep white matter? I would say it mostly primarily focuses on the deep white matter. So this is a little bit of a shameless plug for a review article that is under revision right now at NeuroImage and is already up on arXiv, which is described there at the bottom. We've tried to review all of the different methods for quantitative mapping of structural connectivity using tractography. So it's a long paper with hundreds of citations that we tried to be comprehensive, but one thing that, is kind of clear in the field is that there are two main ways to analyze tractography data. And you can see in the picture on the left, there's the sort of fiber tract oriented method. And in this method, people identify fiber tracts of interest, often based on a hypothesis, and they study something about these tracts, like their fractional anisotropy, for example. Most of these studies focus on the deep white matter because those are the known named tracts that have been of primary focus historically. Some of the studies do include the superficial white matter, but it's more limited. On the other hand, on the right, you can see a connectivity matrix and approaches using the connectivity matrix and the network style analysis of the brain, because they don't do a parcellation or a segmentation in the way of looking for a named anatomical tract. They just take all the tractography data and create the connectivity matrix according to pairwise connectivity between pairs of cortical, usually cortical or gray matter regions of interest. And so they do include also shorter fibers. And so sometimes in various diseases, there can be a result that says the short range connections or the peripheral connections are affected. So if you look deeply, you can find out that yes, there have been more superficial white matter results than we realize in tractography studies, but still it's generally understudied and it's not usually a focus. So we don't know a lot about it yet. Okay, back to our outline. So next I'll shift gears a little bit and I'll talk about the Deep White Matter and Big Data project. In this project, we've recently performed data harmonization and tract extraction and quantification in about 10,000 scans. So we're excited about that and I'll share some of the results. So as probably most of us are familiar, there's new-ish opportunities in these large open data sets that are being released and being acquired. And there on the left, you can see some recent publications that actually are starting to review what are the ethical considerations for the responsible data use and what are the best practices for using this data. And then in the bottom right, you can see that we have a special issue coming out soon in NeuroImage called Advances in Mapping the Connectome, and I'm one of the guest editors of that. There are a lot of cool articles in there that have already come out and some that are in process that deal with new results from these large projects, such as the Human Connectome Project, the ABCD Project, the UK Biobank, and more. So we're in this fascinating era where there are these opportunities to analyze this data. But there are challenges, in particular with diffusion MRI data, big data. So one of the main challenges is that multi-site diffusion MRI data differs across scanners, and the variability of FA can be up to 5% in the white matter or even 15% in the gray matter. And I hear that at this UCLA brain mapping seminar that Yogesh Rathi spoke recently. And so I think he told you a lot about harmonization already. So I'm going to just speak briefly about that. And another challenge is how should we analyze the data? So we're interested in extraction of the anatomical tracts and the superficial connections that we can find, but you really need robust methods and they must be automated if you want to analyze thousands of scans. And so this enables quantification and visualization of important systems that we'd like to study in the brain. Another challenge is just the infrastructure need. You need either a large computational infrastructure at your institution, or you need to use cloud computing just to store and compute to analyze this data. So this is obviously not accessible to all groups who might be interested in analyzing the data. So we have a new, recently, a couple years old R01 that we're working on addressing these challenges and basically providing the data we extract back to the public. So the summary of our ongoing project is this is a multi-PI project. My co-PI is Yogesh Rathi. You can see his picture over there on the right. We have a couple of goals. We plan to harmonize diffusion MRI data across the sites, extract and quantify white matter tracts automatically, provide all of this data back to the public via the NIMH NDA, which is the NeuroImage Data Archive. And our goal is to harmonize several large data sets. As you can see in the table below, so far we've begun with the ABCD data set, and that has 21 sites and approximately 10,000 subjects. So the first technology we're employing is data harmonization technology. And this is just an example result from Suhaila's recent paper. And it's showing the average whole brain fractional anisotropy of the white matter before and after harmonization in comparison to a reference site. So the way this works is there's one site that has good data and a lot of subjects that's chosen as the reference site. And then there's a matching process where there are a certain number of subjects from the reference site and a certain number of and then the same number of subjects matched to subjects from each site that you're interested in harmonizing. So for example, Suhaila is currently using 30 and they'll be matched on characteristics such as age and sex. And I don't know everything that she matches on, but she matches these populations in a reasonable way. And then she learns a mapping between the two scanners to try to remove differences due to differences in magnetic field and homogeneities and other scanner properties. And so what this plot is showing is that it works well. The cyan is the target site before harmonization. So it's site 1 or site 2 before it was harmonized, while the reference, the matched reference data is in red. And then you can see that after harmonization, it becomes this light green color where the whole brain FA of the white matter is indistinguishable from the reference site. So this is nice because once these biases due to site-specific scanner effects are removed, then you can naively pool all of the data and analyze it together without needing to do statistical covariates to correct for it later. And this also enables basically any kind of analysis that you might traditionally do without any sort of later correction. So then the second part is to perform automated tract extraction. And this is Fan's work. You can see his picture there on the right. Our software package for this is called White Matter Analysis, and it's all open source. And there's a website that you can see here at dmri.slicer.org if you're interested in playing with our atlas or applying this technology to any of your projects. And so what you can see on the left is these are example association tracts from our atlas. And you can see the image on the right is in one individual subject. These tracts were automatically identified correspondingly to the atlas. So I'm going to talk a little bit about how we created our atlas and how it works and show some pretty pictures of it. So our atlas is an anatomically curated white matter parcellation. It has several steps to create it. It's initially created using data-driven machine learning and then curated with input from neuroanatomist Nikos Makris. So first we do fiber clustering to group streamlines across subjects according to their similarity in shape and location. And then we worked with Nikos Makris to visually inspect each cluster. For that reason, we limited our number of clusters to 800 in order to make this a feasible process. And he, in conjunction with Fan, performed expert anatomical annotation or labeling of clusters. And so that gave us this example over on the right. So the fiber clustering across subjects to create our atlas, we use data from 100 healthy Human Connectome Project subjects, very high quality data. And we used robust UKF tractography that's also highly consistent across subjects. What you can see on the right is examples of anatomical tracts in the atlas. The one on the top is our representation of the corticospinal tract in the atlas. So each anatomical tract contains multiple clusters. The purpose of the clusters is to do a fine scale parcellation that corresponds across subjects. That's a good representation of what white matter anatomy is present in the group. And then the purpose of basically annotating the clusters is to identify which clusters are part of known named anatomical tracts or regions. So you can see our CST on the right, and on the right, different colors represent different clusters within that particular anatomy. You can see the representation of the SLF2, where again, we have multiple clusters to show we find the SLF2 and also multiple subdivisions of the SLF2 that also might be of interest to study. And you can see on the bottom, the representation of the arcuate in the atlas, including subclusters in there. Another thing that we did here is we identified false positive connections where tractography often would trace basically from one known anatomical structure onto another anatomical structure. And those are identified, so those can be excluded in a study. So you can say, I don't want to include these typical known false positive tractography connections because we know they're not anatomically accurate. You can remove those for your study. So overall, our atlas and our accompanying software is able to robustly identify white matter fiber tracts across the lifespan. We've shown from neonates to elderly, from I think age 2 to age 82 is what we've tested. So across the lifespan, across different data sets acquired at different scanners, across different health conditions, and across different diffusion MRI acquisitions. So this image on the left, again, is an example of 3 tracts that are in our atlas that was learned from the 100 HCP subjects. And then you can see on the right examples, each column shows an example of identifying these corresponding tracts in individual subjects. You can see the column that is an adult from the Human Connectome Project, so it looks very similar to the atlas. Then you can see a neonate. And you can see that those tracts look a little bit different from the atlas. You know, as we might expect, the arcuate fasciculus is very slightly less robustly traced. I mean, language and the white matter fiber tracts are still developing in the neonate. So we would expect to see a little bit of differences. And then, but we were kind of amazed we could identify these. This is a neonate from the Developing Human Connectome project. So very, very high quality diffusion MRI scan of a neonate. And then on the right, you can see a brain tumor patient. This is a scan from Brigham. This is a clinical quality scan with a brain tumor shown in green and surrounding edema shown in gray. And so even in challenging cases, we are able to identify large white matter tracts in the majority of cases. So why is it, working as well as I claim, right? Basically, it's a robust parcellation method. We've compared our method to an alternative, which is parcellating tractography according to a cortical parcellation. And so this is very commonly done, but it suffers from some challenges that there are spatial distortions that aren't fully corrected for between the diffusion MRI and the anatomical T1 and T2 weighted scans used for the cortical parcellation. And also from challenges with tractography that may, in some cases, have difficulty reaching completely, fully, perfectly to the cortex. So we compared our parcellation method using clustering to a parcellation method using basically according to a cortical parcellation. And essentially, we showed that the fiber clustering obtained higher test-retest reproducibility in the same subjects. So as far as a structural parcellation, this gives a sense that it's a highly robust way to do it. And another basically source of robustness in terms of finding all of these tracts in these very large data sets is that we're using a robust method for tractography. We're using the UKF tractography method that was developed by Yogesh Rathi's group, where UKF stands for unscented common filter. And so this method traces through crossing fiber regions. It's a multi-fiber model, and it estimates model parameters consistently along the tract. So it's jointly doing the fiber model estimation and the tractography. So what that means is it has information from the previous step. As tractography takes steps, it has information from the previous step to aid it in fitting the model during fiber tracking and basically deciding where to go next. And so as you've seen in our previous slides, this tractography method in combination with our Atlas fiber tract identification has very consistent results in diffusion MRI data across the lifespan and across different acquisitions. So I have a fun video that I won't show all of. This video is on YouTube, but this shows some of the fiber tracts that are annotated in our atlas, which includes 58 deep white matter tracts and 198 superficial fiber clusters organized into 16 categories according to the lobes that they connect. So this is showing the clustering result across the 100 subjects who were used to basically learn this anatomical parcellation. And these are the annotated clusters that have been grouped into known anatomical tracts with neuroanatomical names. But you can see that each tract has multiple colors within it, which indicates multiple clusters that were grouped to form that tract. So it gives the opportunity to study the tract as a whole, and also to study different sort of sub-connections within that anatomically named fiber tract. Okay. So I'll get back to our current results from this R01 project. So we're doing cloud computing for large-scale analysis of the ABCD, the Adolescent Brain Cognitive Development data set. And so this has almost 10,000 subjects from 21 different sites. And our computational analysis pipeline is fully open source. So if anyone is interested in using it on their data, you can please reach out to us. We're happy to provide more information, or you could, it's open source, it's online. You could use it without telling us if you wanted to, but of course we'd be happy to know. We deployed it on Amazon Web Services. And so far we've done diffusion MRI data harmonization, tract extraction, and quantification of tract microstructure. So multiple measures from the fiber tract. So far we've completed that for this ABCD study. So our initial results are good. We were happy to find out that the diffusion MRI harmonization actually improves the white matter parcellation. So we expected it would stay the same because we know that the diffusion MRI harmonization shouldn't greatly affect the data, but we were happy that it actually improved the parcellation, which is a great result. So after harmonization, we could detect a higher number of anatomical tracts and fiber clusters, and their FA values, as expected, were closer to the harmonization reference data. And we also did successful tract identification across the approximately 10,000 subjects, with a total of 99.9% of the major anatomically named tracts being identified. So I think essentially, if it wasn't identified, it's likely there might have been a problem in the scan. So these figures show the result that the harmonization actually improved our white matter parcellation result. On the left, you can see a bar graph from a fine scale parcellation. So the y-axis, apologies, that's unlabeled. Oh, I guess it's in the title, right? The y-axis shows the identification rate of our 800 clusters in our atlas parcellation. So it basically, you can see there's a 1 on there, which would be perfect 100% identification rate. And you can see that here there are 3 sites, 1, 2, and 3. And the blue is the original. So we tested extraction of the 800 clusters in the original data. And then we also tested it, we ran it, of course, in the harmonized data. And as you can see, at 2 of the sites, there was a significant improvement between the original and the harmonized data in terms of successful extraction of these clusters. And as far as the tract parcellation, these are so much larger than the clusters. They're very robust to extract. And you can see on the right that, while the rates, the identification rate very slightly improved, they're all just about at 1, and there aren't any significant differences, as we would probably expect. This is an example tract visualization of 5 example subjects from this 10,000 subjects, showing some of the tracts that we automatically extracted across subjects. So the take-home message is basically that we've successfully extracted these tracts. And the cool part is that now we can take a look at the data measured from these tracts. Now, this is a neat visualization created by Steve Piper. As part of our grant, we're trying to do web visualization of this data and of the tracts to aid people in performing studies with it. So we're trying to enable neuroscientific studies using this large database without having people have to do all of the data processing on their own at their lab. So as you can see, as he's moving his mouse, it's selecting different subjects. This is the FA on the left across multiple tracts. And as he moves his mouse up and down, it's showing the corpus callosum tract from that individual subject where the FA was measured. And this is part of the posterior corpus callosum, I believe. Okay, so good news is that this data will become available at the NeuroImage, NIMH, National Institute of Mental Health Data Archive, the NDA. This data will all become available online for everybody. So the harmonized diffusion MRI to analyze with whatever pipeline you're interested in, the extracted tracts if you'd like to then analyze the tractography itself, or if you'd like to analyze the individual extracted tracts, and also all of those quantitative measures such as FA and mean diffusivity and more that we extracted from the tracts will be available to analyze. So somebody with a hypothesis could simply download a spreadsheet and actually test their hypothesis without having to process the NeuroImage data directly. Okay, I'm going to move on. Let's see, how am I doing on time? I'm going to move on to the second project. So this is a project that we're studying the brain's superficial white matter, which is much less well known. So I'll talk some about the current state of the art in studying the superficial white matter with diffusion MRI, especially with tractography, and talk about some recent progress and what we're working on. So this is a grant that just started this year. So we're very excited about this new project. Okay, so the superficial white matter is known to be quite important in health and disease. So apart from the fact that it's fascinating because we don't exactly know what's there because we haven't mapped it, it's also very important to learn about to better understand the brain in health and disease. It's particularly important in neurodevelopment and aging. There's a last in, first out hypothesis, where the superficial white matter is the last to myelinate, but also the first to degrade. I'm looking for the right word. The first to have problems during aging. So it's known to be affected in MS, autism, Alzheimer's, and schizophrenia. And shockingly, the superficial white matter represents the majority of white matter connections in the human brain. But it's hard to map due to limited spatial resolution and other challenges. Its anatomical variability is also challenging. The cortex is much more highly variable than the deep white matter tracts, and the superficial white matter being immediately next to the cortex is inherently more variable across subjects than the deep white matter in terms of its anatomy. And this is considered to be a rich, quote, missing data set in diffusion MRI, simply because we can't fully trace it right now or study it with our current technology. So this is the slide where I explain why I call superficial white matter small data. On the left, you see an image from a very nice paper by Dr. Song that was published in 2014. And this is the paper where they show that you really can't see the short cortical association or superficial white matter U fibers in typical 2 millimeter data that you see on the right, in the left image, but the right where it says B. And we've looked, you also can't see these very short ones in even really good, close to 1 millimeter data like the Human Connectome project. You can't fully see these. And then they show with a scan at a higher resolution, sub-millimeter, 0.85 millimeters, suddenly they can trace and visualize all of these shorter U fibers that are near the surface of the white matter. And we have a similar result on the right. This is from my colleague Yogesh Rathi, who's my co-PI, one of my co-PIs on this grant. And he and his group have a very cool method for accelerated sub-millimeter in vivo diffusion MRI. And you can see an example scan there at 0.6 millimeters, and you can see the tremendous difference in U fibers and what we're actually able to view there that we can't see at all in a downsampled 2 millimeter resolution, and we really can't see even in, say, 1.25 millimeters. So it's important to get sub-millimeter data to have a better chance at really more fully depicting these fiber systems. However, there's another challenge here, and this is a joint challenge between deep white matter and superficial white matter as far as tracing and mapping their anatomy. Article came out, I see the date's not there, but relatively recently in the past 5 years or so. And the title of this article is Superficial White Matter Fiber Systems Impede Detection of Long-Range Cortical Connections in Diffusion MRI Tractography. And so their main finding here is when they did very, very, very high resolution, 250 microns, so 0.25 millimeters, so much higher than the previous images you saw on the previous page. When they do imaging of that high resolution in the macaque, suddenly they see these dense white matter zones just beneath the infragranular layers of the cortex, so very dense superficial white matter zones. But then they say that that impedes tracking of the deep white matter or the long range white matter fiber tracts. So they say that actually half of the cortical surface was effectively inaccessible for long range diffusion tracking, because basically the tractography just couldn't get through the dense U fiber zone immediately under the cortex, so the tractography couldn't reach the cortex. And so the paper is interesting because, of course, the first thing you think is, oh, well, but we don't have that problem in our low resolution data. Does that mean that the low resolution, well, lower or even high quality 1 millimeter resolution data that we have now, does that mean that that data is actually better for long range fiber tracking? And they actually argue in this paper that no, it just means that the lower resolution data that we have now is just wrong. So there are these challenges in basically correctly mapping the anatomy of both the deep and superficial white matter tracts that are sort of joint challenges. And how should tractography handle both of those? Even if we have perfect data, how do we do the tractography to adequately trace what's there? So these are challenges. We can't fully map the superficial white matter with current diffusion MRI that's of above 1 millimeter resolution, but all is not lost. So there are lots of ways to actually study the superficial white matter. There are people who use image templates and surfaces to identify superficial white matter voxels and regions. It's important to note that if you study the region of the superficial white matter in this way based on voxels, for example, that these regions also contain information about long-range fiber tracts that terminate in the cortex. As we saw on the previous page, the long-range fiber tracts, or the previous slide, these long-range fiber tracts basically have to get through those superficial U fibers to reach the cortex. So we can always study the region of the superficial white matter, but we need to know that it's not uniquely the superficial white matter. We have partial voluming there. And then there are other methods. Long to medium range superficial white matter connections can be seen using tractography and current diffusion MRI data sets. And I showed some pretty pictures of those before, and I'll talk a little bit more about current results there. So multiple groups have mapped the superficial white matter using templates. There's a really nice human brain superficial white matter atlas paper by Oishi and Susumu Mori's group in 2008, and they defined 9 superficial white matter blades. They call them blades, and then they have 21 subregions. So this is basically a neuroanatomical sort of organization or categorization of regions of the superficial white matter. So that's useful for studying the superficial white matter. And then on the right, you can see there's a paper that says superficial white matter is a novel substrate of age-related cognitive decline. So making the point that it's important to study and understand the superficial white matter in aging. And in this paper, they generated a template of the superficial white matter across the adult lifespan using data from 141 adults from 18 to 86 years of age. So basically they have a template that you can actually access and download and use to identify, to localize the superficial white matter for your study. And then there's an alternative option, which actually came out of the UCLA group, and they've had several papers looking at a surface that represents the gray matter white matter interface, and basically looking at data at these surface vertices, and they're able to study the superficial white matter in that way. So there's lots of ways to quantify information about the superficial white matter that are out there and are successful. And then, of course, what we're interested in right now for my project and for this talk is how do we map the superficial white matter using tractography? And so what are we able to do right now with current good data like the Human Connectome project? So we can trace medium to long range superficial white matter. These are shorter fibers than the traditional named deep white matter tracts, and they can be studied with existing data. And this is the paper that I mentioned briefly before. That's very, very nice. That's a superficial white matter review that came out in NeuroImage last year. And on the image on the right, the top row A and then the second row B, those rows show 2 different atlases from that group. And the second row is showing basically clusters or connections that are not in the top row. And then the third row is showing clusters from our atlas that are not included in either the first or the second rows. So their paper fundamentally showed there's some overlap with these atlases in terms of what connections are defined, but also that they're very different across atlases. And in part, this is because they were created using different computational methods. And in part, this is because they were created using different resolutions of diffusion MRI data. So ours was generated with a higher resolution, and we can see that that does let you trace basically more connections. So this is a really interesting paper by Pamela Guevara's group that came out again last year. And this is a picture on the right of variability of the superficial white matter across subjects. And basically each row is a subject, 1, 2, 3, and 4, and each column is a connection that they've tried to identify in a corresponding fashion across subjects. And what you can see is if you look at these connections, for example, the orange one, which is the second column, looks quite different across subjects. And then if you look at the red connection, they were unable to find that in subject 4 on the bottom row. So there's definitely anatomical variability that's really interesting and understudied and not really well characterized yet. And so I mentioned this project looked at fibers that were over 30 millimeters in length. So these are not the shortest, most highly variable that we hope to be able to see with the higher resolution diffusion MRI. So this is a overview of our current atlas, which has 198 long-range superficial white matter bundles. So these are even longer than discussed in the previous paper. These are 40 millimeters and longer. So they're really quite medium to long range superficial white matter. This is not the most superficial of superficial, but we've shown that we can find these robustly across subjects, age ranges, and scan protocols. So this is a visualization of finding one particular magenta superficial white matter cluster from 1 day old to 82 years old. I'm actually going to skip the video of the fiber clusters because I want to leave time for questions, but we have a video online, dmri.slicer.org. You can find it if you want to see some of the fiber clusters and especially some of the superficial white matter fiber clusters. I'm also going to skip the challenges and get to some of what we're working on in our project to try to leave time for questions. So we're trying to improve mapping of the superficial white matter connectome using a combination of high resolution diffusion MRI from Yogesh, anatomically informed fiber clustering led by my group and in collaboration, obviously, with Yogesh and with Nikos Makris, who's a neuroanatomist. His picture is on the right, and his fantastic group is going to do basically neuroanatomy-based curation and annotation with reference to all of their knowledge also about non-human primate superficial white matter and existing studies of that. And eventually, at the end of the project, we'll provide all of the data back to the public via the NDA. So our first goal is to try to map previously invisible superficial white matter. And I spent quite a while before talking about this image, and you can see that I like this image. It's really cool that Yogesh provided it and that we are very excited about getting going and being able to get more scans like this. Our second goal is to work on superficial white matter fiber clustering. And so we're really hoping to improve our current machine learning methods to better handle the challenges of the superficial white matter. And I'm going to keep going a little bit because I have two example results. I think I just have 2 or 3 slides left. One thing that we're working on is trying to leverage more information for our clustering. So we know that multiple information sources are good for cortical parcellation. But most tractography parcellation uses either points along the streamline trajectory or region of interest information about what anatomy it passes through. And we're trying to basically design methods that allow us to incorporate multiple sources of information. So this is a neural net here on the right from our Mackay publication from this year that you can see the citation on the bottom. And you can see the arrows. The take-home point is that the arrows, we have trajectory information that's able to be input here. We have anatomical information that's influencing the clustering. We also have a way that we can incorporate pairwise information about the similarity that we expect 2 different fibers to have. So this is what we're exploring and trying to improve. And we're also working on deep learning for superficial white matter parcellation. So this is a recent result that we just submitted to ISMRM. One of my students basically is developing a deep learning method to apply our current superficial white matter atlas, but to better identify the actual structures. And you can see that these are results from 3 data sets, subjects from the HCP, the ABCD, and the PPMI data set. And our new method is in red. And essentially the take-home message is that the red bar is higher than the other bars, but also that there's variability across data sets and methods. Our old method is white matter analysis. And I didn't say that the y-axis is the identification rate for clusters. So for example, in the HCP, we're identifying, maybe over 95% of the clusters that are in our atlas, but then the superficial WMA, which is in red, the new method, the deep learning method, is able to identify closer to 97 or 98%. I don't know the exact numbers. And it's much more dramatic in the ABCD data set that the red bar is quite a bit higher than the others. So actually a little surprising, not only the definition of the clusters is important, but how you find them is important. And finally, our last goal of our current project that we began this year is to work closely with neuroanatomists. We're working with Dr. Edutarian, who has a long history of tracer studies in monkeys during his PhD and during his time as a professor. And this image on the left is from one of his papers, a tracer study. And then working with Nikos and his group with Jarrett Rushmore, who's a neuroanatomist at BU, and basically looking for correspondences between the macaque tractography, the human brain tractography, and what is known about the superficial white matter from existing tracer studies. So we're doing some comparative work as part of this grant to basically evaluate the data that's in the atlas that will generate of the superficial white matter. So in conclusion, a lot of progress has been made for mapping and studying the deep white matter and the superficial white matter using tractography. There are still unique challenges, technical and anatomical, for the deep and superficial white matter, and the joint challenge that one impedes the other in good data for tracing. We can robustly identify deep tracts and longer range superficial connections across the lifespan to enable studies. We're very excited about being able to analyze new, very large data sets. And finally, next generation high resolution diffusion MRI is expected to enable mapping of previously invisible superficial white matter connections. So all of our technology is open source. It's Slicer DMRI for visualization, the white matter analysis machine learning pipeline, and the UKF Tractography. I do have a postdoc position available for the new grant about computational mapping of the superficial white matter. So please spread the word. And if you're interested, definitely contact me. My email is right here, o'donnell@bwh.harvard.edu. And finally, I'd like to thank all of you for coming to the talk, and thank UCLA for inviting me, and thank all of the people who did so much work on all of these projects. Thank you.
Excellent talk, Lauren. Thank you very much.
Thank you.
I have a few questions, but so I see a few questions in chat. Maybe it's best if you read them one by one and answer. Okay,
let me click that button. Yeah. Okay, when you say let me start here. Hypertoxicity. Why is this superficial white matter first to degenerate? I actually don't know. I'm not an expert in aging of the brain specifically. I do know that people who are more expert in this basically say the degeneration begins there, but I don't know the biological underpinnings of that right now. So I think my colleague, Andrea Remia, who studies aging, would be a great person to ask about that. Okay, next is when you say X% identification, how is the 100% reference determined? Okay, so we basically know how many clusters we have in the atlas, and we know how many fiber tracts. There's 800 clusters, and then there's the 58 deep white matter fiber tracts and all of the superficial clusters. So if we identify, 798 clusters, then there's 2 of the 800 we didn't identify. So whatever 90% that is. So it's basically just, we know all the clusters that we identify in an individual subject correspond directly to the atlas. So it's really easy to say whether or not we found a particular one. Very cool talk. Thank you. Is there estimate? Yeah. Is there any estimate of when it will be available on the NDA? Yeah, that's a wonderful question. I wish I knew the answer, because right now we're at the point that it depends a bit on the NDA. We're having meetings with them. We're also developing this visualization tool. And so the path of integrating that with the NDA is slightly less clear. So right now, what we don't know is, will we be waiting for the integration of that? Will we directly upload and allow things for download? Basically, we're ready to upload things, but we're still waiting for kind of some best practices guidance from them to move forward. But if you want more particular details, or you kind of want to be notified when it happens, you can email me directly to be posted. Oh, somebody said excellent. Thank you. Oh, thank you. That's a nice non-question. Thank you.
And if anyone has any other questions, please unmute yourself and ask directly. So while people are thinking, the techniques and the ideas for extracting these short range superficial white matter tracts.
Yeah.
And I don't know if my thinking is correct or not. Maybe correct me on this. And the ideas for extracting very short range fibers in the deep cortical matter, let's say in the hippocampus or the thalamus, and the connections between them. I don't know if you had any insights on that. Is the second problem
it's maybe 2 different questions. So one question is about the superficial white matter and the short connections, and the other one is about short connections that are deeper between, like, subcortical gray matter, for example.
So the question was, are these techniques comparable? Can you bring the same ideas to bear to solve these problems?
In part, yes. So the superficial white matter has some unique challenges because the U fibers are very highly curved. And we know that highly curved structures are inherently challenging for tractography because it sort of wants to go, straight, I mean, going straight is easiest, right? So your model has to accommodate that curvature, and your voxels have to be small enough that the data can actually lead to tracing that curvature. I don't believe that the connections between subcortical gray matter structures are that curved. There wouldn't be a good reason for them to be that curved. An example of a high curvature is like the anterior part of the optic radiation that curves around the ventricle, and that's a known challenge for tractography, and there's many dedicated algorithms designed to track that. So as far as tracing the shorter connections, for most of these atlases, these sort of clustering things, or these sort of automated tract extraction. So for clustering, for example, there are so many short fibers that in order to tune your algorithm to be able to find and depict the long fibers very well, especially sort of historically where we had much less memory and processing power than we do now, you really had to have a length cutoff and just pay attention to what people were most interested in studying. But now it's coming up, we're getting questions from everybody. Is this shorter connection in your atlas? Is this shorter connection in your atlas? We're like, sorry, we sort of followed historical precedent when we had bad computers and we couldn't really handle clustering everything. So I don't think it's necessarily a tractography problem. It's maybe more of our machine learning couldn't handle it before a problem that obviously is not so much of a problem today. And we need to kind of move forward. One of our goals is really our next atlas needs to basically be created on a much bigger machine, because we still want to capture the anatomical variability across subjects, but we'd like to also be able to include a higher proportion of the total fibers we can trace. Now, if they get too short, then they're probably noise. But you know, you don't want something that connects two voxels because you have no sense of that's a meaningful connection. It's like your tractography just couldn't get going. It wasn't like the region wasn't amenable to tracing, sort of thing. But that's a different question.
Okay. So can you bring these shape priors, for instance, these curved, highly curved U connections? So there's a shape in there. These techniques are, I guess maybe the ridgelets. I don't know if it's bringing in shape priors to help the extraction.
I think initially we're just going to try the higher resolution data, because our preliminary results seem to indicate that if you just improve the diffusion MRI data by, you know, by a lot, then you're actually able to trace them. Now, are you able to trace everything? Do you need improved priors to better trace things in the high resolution data? We don't know that yet. So maybe, I mean, yeah, we're open to trying to improve the tractography for that.
Okay.
And we are interested in priors in order to try to trace these in lower resolution data sets once we figured out what they look like in higher resolution data sets. That's a goal in the grant, because what can we develop from this knowledge that's then useful to analyze other data sets that exist? Yeah.
Okay. Any other questions? So when you say these AWS, so all the data, I think one of the goals in your project was to make it available on AWS.
Yeah, on the NDA, which is the NIMH data archive, and that is actually, they do have all their data in buckets on AWS. So part of the motivation for processing it there is you can directly access their data bucket, process it, and in theory, give it back to them in the data bucket that they tell you to.
So for researchers, we have to purchase cloud credits on AWS and then run.
Not to use, well, it depends on what the goal is. If you want to download the measures, the extracted tract measures, and have some hypothesis about, I don't know, the arcufaciculus and the ABCD, you basically just have to go download our spreadsheet and then download the ABCD spreadsheet that gives you whatever demographic or cognitive or other characteristics you're interested in. If you choose to want to basically analyze our harmonized diffusion MRI data, say you want to run, I don't know, TBSS on 10,000 subjects, then it would be recommended to get an AWS account and do the processing there, because then you avoid having to download all of the 10,000 subjects to some other high performance computing center, which is not what you want to do.
Okay. Yeah.
Probably not what you want to do. I don't know.
Yeah. Good point. Cool. Okay, so I just wanted to say something. I shout out to Owen Phillips's work on this surface-based.
Yeah, you're one of the co-authors of that, I saw.
And so Owen is from Kathleen Nahr's group, was from Kathleen Nahr's group. Now he's in Silicon Valley running some company. So
cool.
The superficial white matter studies, which we did, and we applied to many other data sets. So it was robustly replicated across disease populations with a very huge effect size all over the white matter boundary, and which was surprising at the same time intriguing to us. So I guess with the tractography approach, which of course is the high resolution, the right anatomical approach, hopefully we see something more sensitive as opposed to more diffuse.
Hopefully. I mean, you have the advantage also that you can have your model try to be specific to the tract and try to separate out partial voluming of other crossing tracts trying to insert in the cortex. So, you know, ideally, you're able to have a chance of studying something, you know, is it the superficial connections that are having a problem, or is it the location where the other ones are crossing that are having a problem? You could at least aim to shed some light on that and perhaps have more specificity to what's being connected and what's the trajectory and the location. But again, that's our hope.
Yeah. Okay. Great. So if there are no more questions, let's thank Lauren again, and happy holidays, everyone, and see you guys next year. And then Lauren, this sparked a lot of interesting discussion and some new questions as well. So I guess.
Thank you very much. Yeah, I'm happy to chat more actually, if you think that would be interesting offline another time.
Yes, sounds good. Sounds good.
Thanks.
All right. So this is the end of the seminar. Happy holidays, everyone. See you next year.
Thank you.